Okay, today I will start this second unit, nucleic acids and lipids. Okay, let us see a brief introduction about nucleic acids. Do you have any idea about nucleic acids?
Nitrogen, sugar, and they contain phosphate or any other group.
Okay, very good, very good. So you are saying that the composition of nucleic acids, right? Okay, let us see. Nucleic acids. There are two types of nucleic acids. They are deoxyribonucleic acid, DNA, and ribonucleic acid, RNA. So there are two types of nucleic acids present in the human cells, DNA and RNA. Okay. What is the role of these nucleic acids? Okay, human cell contains two types of nucleic acids, DNA and RNA. Then what is the role of these nucleic acids? These nucleic acids helps to store and transmission of genetic information from parents to children. So these nucleic acids play key role in storing and transmission of genetic information. And they also helps to give instructions to make proteins and enzymes for various metabolic cellular functions. See, what is the role of these nucleic acids? DNA and RNA, what are the uses? ఏం చేస్తాయి అవి? They play a key role in storage and transmission of genetic information from parents to children. Okay, and they also provide some instructions for making proteins and enzymes for various types of cellular functions. Okay, now where they are located? Okay, human cell has contains DNA and RNA nucleic acids, but ekkaduntayi mari? See this figure, and this is a cell structure, and you can see the round one. The center of the cell is called nucleus. Okay, and this red one is called cytoplasm. There are other parts, but the main is cell wall, cell, and the center of the nucleus cell is called nucleus. Okay, if you see, the center of the nucleus has some thread-like structures. See here, center of the nucleus has some thread-like structures. These are called chromosomes. The thread-like structures are called chromosomes. Okay, so here the DNA, deoxyribonucleic acid, is present in the chromosomes of the nuclei. Chudandamma, nuclei. This nucleus has thread-like structures called chromosomes. These chromosomes contain large amount of DNA along with histone proteins. So what is the where is the location of DNA? DNA is present in the chromosomes of the nuclei, right? DNA is present in the chromosomes of the nuclei. Chromosome is composed of large amount of DNA and inka emuntayi histone proteins. Okay, so DNA is present in the nucleus but in chromosomes. And RNA is located in the cytoplasm of the cell. RNA is not present in the nuclei. RNA is present in the cytoplasm of the cell. Okay, so there are two types of nucleic acids in the human cells, DNA and RNA. DNA enti name cheppandi? Deoxyribonucleic acid. RNA is ribonucleic acid. What is the role of these nucleic acids? They play a main role in storage and transmission of genetic information, and they also give some instructions for making proteins and enzymes for various cellular functions. And DNA is present in the chromosomes. RNA is present in the cytoplasm of the cell. Okay, this is the very brief introduction about DNA and RNA. Let us see the composition of DNA and RNA, how they are made up of. అందులో ఏమేమి ఉంటాయి చూద్దాం. See, we can say a polymer chain of nucleotide is called a nucleic acid. We can say a polymer chain or a chain of nucleotides is called nucleic acids. That means every nucleic acid is made up of a chain of nucleotides. So the monomer unit is nucleotide amma. A chain of nucleotides is called nucleic acids, or a polymer chain of nucleotides is called nucleic acids. And nucleic acids are composed of nucleotides. nucleotides chain anta కలిపి మనం ఏమంటున్నాము nucleic acids. Okay, a chain of nucleotides is called nucleic acids. Then what is nucleotide? See, chain of nucleotides is called nucleic acids. Then what is nucleotide? Nucleotide is composed of three components: nitrogen base, pentose sugar, and phosphate group. Each nucleotide is composed of nitrogen base, pentose sugar, and phosphate group. అర్థమైంది కదా? Nucleic acids are polymers of nucleotides, or a chain of nucleotides is called nucleic acids. And every nucleotide is composed of three components: nitrogen base, pentose sugar, and phosphate group. Let us see this nitrogen base. Okay, ఇప్పుడు ఈ components గురించి చూద్దాం. Nitrogen bases. See here, nitrogen bases. It's a component of nucleotide. There are 5 nitrogen bases in DNA and RNA. అన్ని కలిపి చూస్తే nitrogen bases are 5 ఉన్నాయి ఇక్కడ, 5 types: adenine, guanine, cytosine, thymine, and uracil. There are 5 nitrogen bases in DNA and RNA, but each nucleic acid contains only 4 nitrogen bases. Please remember, total number of nitrogen bases are 5: adenine, guanine, cytosine, thymine, and uracil. But DNA contains 4 nitrogen bases, RNA contains 4 nitrogen bases. అవి ఏవేవి అనేది చూద్దాం. See here, adenine and guanine. Adenine we can denoted by A. Guanine can be, symbol is G. Okay, this adenine and guanine are derivatives of purine. Chudandamma, these adenine, guanine, cytosine, thymine, and uracil, these nitrogen bases are actually heterocyclic aromatic amines. They are heterocyclic aromatic amines. You can see the structures here also. In this, adenine and guanine are derivatives of purine. ఇందులో adenine and guanine అనేవి ఏంటట? Purine derivatives. Let us see the purine structure. See here, this is the structure of purine. This is the structure of purine, and it is a bicyclic structure, and it is a heterocyclic amine. See, all nitrogen bases are ఏమన్నారు ఇక్కడ? Heterocyclic aromatic amines, and this purine is heterocyclic aromatic amine. It is made up of 2 cyclic rings, right?
Excuse me, ma'am. Ma'am, some people are waiting for you to join.
Who is waiting?
They join, ma'am.
Okay, purine is a heterocyclic aromatic amine. You can see the structure of purine here. ఇక్కడ మీరు చూడొచ్చు purine structure. It is a bicyclic structure. What is this? What is the name of this left side ring? Please unmute yourself and give an answer. What is the name of this left side ring?
Benzene.
Please observe carefully. Andaru vinandi. This is the purine structure. Okay, ఇది purine structure. It is a bicyclic structure, means there are 2 cyclic rings. What is the name of this right side ring? This one. I am moving cursor, you can see. It ఏం చెప్పండి అమ్మా, రైట్ సైడ్ రింగ్ ఏంటిది? 5 membered ring ఉంది చూడండి, NH, and this one. ఎవరో చెప్పారు, నేను వినలేదు. ఒకసారి ప్లీజ్ రిపీట్.
Imidazole.
Very good, very good. See, pyrimidine and imidazole. There are 2 types of moieties: pyrimidine and imidazole. These 2 combination is there in purine. Okay, this is the pyrimidine ring and this is imidazole ring. Okay, because you have to remember this structure. అర్థమైందమ్మా? మీరు ఆ ఎగ్జామ్ లో రాయడానికి మీకు గుర్తుండాలి కదా. సో దిస్ ఇస్ ద స్ట్రక్చర్ ఆఫ్ ప్యూరిన్, అండ్ యూ కెన్ సీ ద నెంబరింగ్. సి దిస్ 6 membered ring, you should start numbering from large membered ring. So we are going to give the numbering from this nitrogen: 1 2 3 4 5 6, then 7 8 9. You have to remember this. Sometimes they will give 9 to 7 8 9. NH will get 7 also, because both are in equilibrium tautomers. So you can give this 1 2 3 4 5 6 will be same, but sometimes you will see this nitrogen 7, and sometimes you will see this nitrogen 7. Okay, but the overall structure is like this. This is the purine. Okay, ఎందుకు ఇదంతా చెప్తున్నాను అంటే, adenine and guanine nitrogen bases are derivatives of purine. See this, adenine and guanine, these 2 nitrogen bases are derivatives of purine. See, they have similar structure. See, this is purine moiety, and this is also purine. But in adenine, additional substance, what is the additional substance? చూడండి అమ్మా, ఇక్కడ ఇదంతా ప్యూరిన్ స్ట్రక్చర్ అడెనిన్ లో, బట్ ఎక్స్ట్రా సబ్స్టెంట్ ఒకటి ఉంది. వాట్ ఈస్ దట్? అమైనో గ్రూప్. వెరీ గుడ్, అమైనో గ్రూప్. సో ఇఫ్ యూ ట్రై టు గివ్ ద నేమింగ్, చూడండి నెంబరింగ్ ఇస్తే ఏమవుతుంది? 1 2 3 4 5 6th ప్లేస్, అవునా? 6 అమైనో. 6 మెంబర్ రింగ్ కి నెంబరింగ్ ఇవ్వాలమ్మా ఫస్ట్, తర్వాత 5 మెంబర్ రింగ్ కి. సో 1 2 3 4 5 6, 6 అమైనో adenine. Okay, see this one, 6 amino adenine, right? And guanine, cheppandi, 1 2 3 4 5 6, and this one 2, this one 1, this one 3. So 2 amino 6 oxopurine, 2 amino 6 oxopurine. So these two nitrogen bases are derivatives of purine. Chudana, adenine and guanine are derivatives of purine, and remaining cytosine, thymine and uracil are derivatives of pyrimidine. I think someone is trying to join. Let us see. అర్థమవుతుంది కదమ్మా? అడెనిన్ అండ్ గ్వానిన్ ఆర్ డెరివేటివ్స్ ఆఫ్ ప్యూరిన్, అండ్ రిమైనింగ్ సైటోసిన్, థైమిన్ అండ్ యురాసిల్ ఆర్ డెరివేటివ్స్ ఆఫ్ పిరిమిడిన్. సీ దిస్, యు నో దిస్ పిరిమిడిన్ స్ట్రక్చర్, అవునమ్మా? సో దీస్ నైట్రోజన్ బేసెస్, థైమిన్ నైట్రోజన్ బేస్, సైటోసిన్ నైట్రోజన్ బేస్ అండ్ యురాసిల్ నైట్రోజన్ బేస్ హ్యాస్ pyrimidine moiety. చూడండి, n and alternate nitrogens, alternate nitrogens, alternate nitrogens. So these three nitrogen bases have pyrimidine moiety. So these are pyrimidine derivatives. Adenine and guanine has purine moiety. So these two are purine derivatives. Okay. And now the most important one is DNA is made up of, or DNA is composed of 4 nitrogen bases. DNA aina RNA aina kuda they contain only 4 nitrogen bases. But what is the combination? Chuddam. DNA is composed of adenine, guanine, cytosine, thymine. DNA lo ఏమేమి ఉంటాయమ్మా? అడెనిన్, గ్వానిన్, థైమిన్, సైటోసిన్, దీస్ 4. అడెనిన్, గ్వానిన్ purine derivatives, thymine, cytosine pyrimidine derivatives. So DNA has 2 purine derivatives, 2 pyrimidine derivatives. What are the names? Adenine, guanine, cytosine and thymine. Okay, next RNA. RNA also has 4 nitrogen bases. What are the names? Adenine, guanine common ga untayi renditlalo. Adenine, guanine, and remaining 2 ఏంటి చూడండి, cytosine and uracil. అంటే just only one difference, thymine and uracil are differ in these 2 nucleic acids. చూడండి అమ్మా, adenine, guanine, cytosine are common in both nucleic acids, అవునా? But in DNA thymine is there, but in RNA in the thymine place uracil is there. Okay, this is about nitrogen bases. మీకు సింపుల్ గా మీరు ఇలా గుర్తుపెట్టుకోండి, పిరిమిడిన్ డెరివేటివ్స్ అనగానే కట్ గుర్తుపెట్టుకోండి, కట్ చేయండి, కట్, CUT, C cytosine, U uracil, T thymine. Okay, ఈజీగా గుర్తుంటుంది. నెక్స్ట్, see here, nucleotide is composed of 3 components: nitrogen base, pentose sugar and phosphate group. Okay, now we have discussed already about nitrogen bases. Let us see this pentose sugar. Pentose sugar, see this pentose sugar ఎందుకంటున్నాం చెప్పండి?
5 carbon sugar.
Very good, very good. So this sugar has only 5 carbon atoms. That is why we are calling this pentose sugars. And especially we can call it as aldopentoses. Aldopentose sugars antam. Aldopentose sugars అనడానికి కారణం చెప్పండి?
Aldehyde group.
Very good, you are very perfect. Okay, so these sugars or monosaccharides are called as pentose sugars or aldopentose sugars. Why they are monosaccharides? Why
only one sugar molecule?
See, it's not a trimer or polysaccharide, so they are simple monosaccharides. So nucleic acids contain monosaccharides, and what is the name? Pentose sugars. Specifically em cheppochu? Aldopentose. Okay, so why we are calling pentose sugars? Because of 5 carbon atoms. And this is, chudandi, I am showing this type of representation. Can you guess what is the name of this representation? ఇలా రాసాను కదా skeleton type, skeleton structure. I'm here you are seeing skeleton like structure. The
Fisher
name
projection.
very good. Fisher projection representation. ఇలా రాస్తే ఏమంటాం మనం దీన్ని? Fisher projection. Okay. So now let us see which sugar is present in RNA, which sugar is present in DNA.
Ribose sugar present.
Yeah.
RNA.
We know ribonucleic acid. So ribonucleic acids, first one R is ribose. So ribose sugar is present in RNA. Next one, DNA. D means deoxyribose. Deoxyribonucleic acids. That means deoxyribose sugar is present in DNA. That is why the name is deoxyribonucleic acids. Okay. See, the sugar present in the nucleic acids are aldopentoses. A pentose sugar have 5 carbon atoms. And the sugar component of RNA is D-ribose. Why we are indicating this D here? Hydroxyl.
Conformations.
Yeah, DL nomenclature. According to DL nomenclature, here this 4th carbon hydroxyl group is on the right side. Okay, so D-ribose. ఇలా ఉంటాయి అన్నమాట. So in the RNA, D-ribose is present, or simply we can write ribose sugar. And in the sugar component in DNA is called 2-deoxyribose. Okay, 2-deoxy. Why we are calling it as deoxyribose? Stephanie, this is
Less
Oxygen is absent in the
second carbon.
absence of
oxygen atom on the second carbon atom.
Second carbon.
very good. So due to the absence of OH, or due to the absence of oxygen at second carbon atom, in the ribose we can see hydroxyl group at second carbon atom, but in the 2-deoxyribose we can see there is no oxygen atom, just hydrogen is there. So that is why the name is 2-deoxyribose. Only that is the difference between RNA and DNA. That is the main difference. So based on that difference we can distinguish these nucleic acids in different organisms also. Okay, this is about sugars, or monosaccharides or pentose sugars. Next one, what is the third component in nucleotides?
Phosphate.
Phosphate group, right?
Yes.
The first component is nitrogen base, then pentose sugar, and then phosphate group. Let us see. Okay, I have to tell one thing. Sorry. Let us see the formation of hemiacetal. See this. This is D-ribose, and this is 2-deoxyribose. This ribose is present in RNA, and this ribose is present in DNA. Manaku telsu ippadi varaki. Okay, but in the human cell, in the nucleic acids, these ribose and 2-deoxyribose sugars are present in the cyclic form, or anomeric form, or furanose structures. Ila straight ga undadu DNA RNA లలో. It appears in cyclic form. They are in equilibrium with this straight chain, and these cyclic structures are in equilibrium. Okay na ma, manaki DNA RNA lo ila cyclic structure lo untayi arrangement anedi. Okay, so this ribose and 2-deoxyribose on cyclization gives hemiacetals. Okay, someone is joining. Let us see. See this D-ribose, first one is judanama, D-ribose on cyclization. This hydroxyl group attacking on this aldehydic carbon. That is why we are getting cyclic structures. We can call them hemiacetals, or ribofuranose. We can call it as ribofuranose. I think you might have studied in carbohydrates topic. Hexoses like aldohexoses can form pyranose structures. Avuna? Aldohexoses can form pyranose cyclic structures. But this is pentohexose, aldopentose. So only five carbons are there. 5 carbon member ring nemantam manamu ante furanose. Gurtu pettukondi. If you have glucose here, the cyclic hemiacetal is called pyranose. But here we are taking this ribose sugar on cyclization by the attacking of OH group at 4th carbon to aldehydic first carbon gives cyclic structures called hemiacetals, or anomers, or furanoses. And in this hydroxyl group, if this hydroxyl group at first carbon is on right hand side, we can give the name alpha. Chudandi, if hydroxyl group at first carbon, this is anomeric carbon. Okay, anomers ante enti? Anomers are the cyclic saccharides. Okay, cyclic saccharides. Cyclic structure lo unnai. Iventi anomers. So this is anomeric carbon. If OH group is at right side, the name is alpha. Gurtupetukondi. Alpha D ribofuranose annam. Why we are calling furanose? Why we are saying furanose? Because of 5-membered ring. 6-membered ring aithe emanali meeru pyranose anandi. And 5-membered ring, see 1, 2, 3, 4 and including oxygen 5. So these are 5-membered ring structures. That is why the name is furanose. So if OH group is on right side, alpha D ribofuranose. If OH group is on the left side, the name is beta D ribofuranose. Okay, and this is alpha anomer and this is beta anomer. In the nucleic acids, these ribose sugar and deoxyribose sugars exist in the beta form. Nucleic acids lo ivi A form lo untayi, beta form lo. So in the Haworth structures, you have to write this hydroxyl group on upside. See here, beta anomers lo eppudaina OH group paiki rayandi. In the beta anomer, this OH group is on the upside. And if you see this alpha anomer, this first carbon hydroxyl group is represented at downside. So this is the difference. Here I am showing this Fisher projection formulae, Haworth structures and Haworth wedge dash projection formulas. See what is the difference between alpha anomer and beta anomer. In the alpha anomer, hydroxyl group is at right side of the first carbon. And what is beta anomer? Here OH group is at the left side. But nucleic acids lo meer eppudu rasina kuda we will represent the structures in this format. So please remember beta D ribofuranose or beta anomer ante enti. Meer eppudu rasina kuda you have to write this OH group is on above direction or upside direction. So this is beta anomer. And remember, in the nucleic acids, this ribose sugar exists in the beta anomeric form, not like this. You can see only this beta anomeric form. Similarly, in 2-deoxyribose in the DNA, this 2-deoxyribose on cyclization forms similar type of hemiacetals. What is the difference? Here, there is no oxygen in the deoxyribose. Here also, if hydroxyl group is on right side, we can call it as alpha anomer or alpha 2-deoxyribofuranose. And this OH group is on left side. What is the name? Beta 2-deoxyribofuranose. And what is this? Beta anomer. And here you can write, you have to practice these structures. See this. In the DNA also, this nucleic acid, in the DNA also, this sugar exists in the beta anomeric form. That is why you should write this OH on above side, above direction. So, and this is second carbon. See this, the difference. Here second carbon has hydrogen and OH. Second carbon has hydrogen and OH. In the deoxyribose, second carbon has only 2 hydrogens. That is why we are writing 2 hydrogens. This is only the difference. So, in the hemiacetal formation, what is happening? This hydroxyl group from 4th carbon attacking on aldehydic carbon. So, we are getting hemiacetals. There are 2 types, alpha anomer, beta anomer, or alpha D ribofuranose or beta D ribofuranose. But among these, this beta anomer is appear in the nucleic acids. Okay? See, just remember these 2 structures. Just remember these 2 structures. This is the beta anomeric form of ribose. See, in nucleic acids, sugars are in furanose form or beta anomeric form or hemiacetal. Edaina okate. But alpha kaadu gurtupetukondi, beta anomeric form. Okay. Next, the third component is phosphate. What is formula of phosphoric acid? Cheppandi okkasari. What is the formula of phosphoric acid?
H3PO4.
H3PO4. So H3PO4 can form different types of anions, right? If it loses only H plus ion, dihydrogen phosphate. If it gives 2H plus, if it loses or dihydrogen phosphate loses H plus ion and gives monohydrogen phosphate. Monohydrogen phosphate, if it loses H plus ion, it gives phosphate, simple phosphate antam manam. Dihydrogen phosphate, monohydrogen phosphate, and phosphate groups. Okay, this phosphate group is present in the nucleic acids, and this phosphate group and the sugar monosaccharide and nitrogen base, these three combinedly to form nucleic acids. And you can see this, చూడండి ఇక్కడ న్యూక్లిక్ యాసిడ్స్, చైన్ ఆఫ్ న్యూక్లియోటైడ్స్. అవునా, న్యూక్లిక్ యాసిడ్స్ అంటే ఏంటమ్మా? చైన్ ఆఫ్ న్యూక్లియోటైడ్స్ ఆర్ పాలిమర్స్. న్యూక్లియోటైడ్ పాలిమర్స్ ఆర్ కాల్డ్ న్యూక్లిక్ యాసిడ్స్. ఎలాగైనా అనొచ్చు, నంబర్ ఆఫ్ న్యూక్లియోటైడ్స్ కలిస్తే వచ్చేది మనకి న్యూక్లిక్ యాసిడ్స్. నౌ వాట్ ఈస్ న్యూక్లియోటైడ్? It is the combination of three components: nitrogen base, sugar and phosphate. ఇంతవరకు మనకు క్లియర్ గా అర్థమైంది. Nitrogen base, sugar and phosphonate, these three combination is called nucleotide. If you take only this nitrogen base and sugar, combination of nitrogen base and sugar is called nucleoside. గుర్తుపెట్టుకోండి. సో నైట్రోజన్ బేస్ అండ్ షుగర్ రెండే తీసుకున్నారనుకోండి, ఏమంటారు దాన్ని? న్యూక్లియోసైడ్. అడినిన్, గ్వానిన్ ఉన్నాయి కదా, నైట్రోజన్ బేస్ ప్లస్ షుగర్, దిస్ టూ కాంబినేషన్ is called nucleoside. Nitrogen base, sugar and phosphate group, these three components combination is nucleotide. The combination of the repeating units of these nucleotides is called nucleic acids. Number of nucleotides kaliste vachedi nucleic acids. Nitrogen base, sugar and phosphate group kalipite vachedi nucleotide. ఇందులో మళ్ళీ సింప్లిఫై చేయండి. నైట్రోజన్ బేస్ అండ్ షుగర్ ఈ రెండు యాడ్ చేస్తే మనం ఏమంటాం దాన్ని? న్యూక్లియోసైడ్. ఓకే, లెట్ us see the types of nucleosides. What is nucleoside? చెప్పండి ఇప్పుడు. What is nucleoside?
Sugar plus nitrogen base.
Very good. The combination of nitrogen base and sugar is called nucleoside. See, we have two nucleic acids, RNA and DNA. So we can classify these nucleosides again into two types: ribonucleosides, deoxyribonucleosides. RNA గురించి చదువుకున్నప్పుడు ఏమంటాం వాటిని మనము? రైబోన్యూక్లియోసైడ్స్. DNA లో మనం కాంబినేషన్ చూస్తే ఏమనాలి? deoxyribonucleosides. ఓకేనా? సో see this, if adenine is taken, if you take this adenine and one sugar, the name is adenosine. The nucleoside of adenine, చూడండి, combination of pentose sugar and adenine, this is adenine amma. Combination of sugar and adenine is called adenosine. The name of this nucleoside is adenosine. If you take guanine తీసుకున్నారనుకోండి, for example, sugar is common. Okay, if you take sugar and if you take guanine, the name is guanosine. మామూలు నైట్రోజన్ బేస్ చదువుకున్నప్పుడు మనం అడినిన్, గ్వానిన్, సైటోసిన్, థైమిన్ అన్నాం. న్యూక్లియోసైడ్ గా మారినప్పుడు దాని పేరేంటి? వెన్ అడినిన్ ఈస్ కంబైన్డ్ విత్ దిస్ షుగర్, ద నేమ్ ఈస్ అడినోసిన్. ఓకే, అండ్ దిస్ ఈస్ గ్వానిన్. Guanine and sugar is called guanosine. But here see, some difference is there because the nitrogen base name is cytosine. Sorry, nitrogen base name is cytosine. This is cytosine. That is why here the combination is given as cytidine. And this is uracil, nitrogen base. Combination of uracil and sugar, nucleoside name is uridine. Just remember, adenosine, guanosine, cytidine and uridine. These four are ribonucleosides. RNA lo uracil untundani chadukunnam kada. RNA is composed of adenine, guanine, cytidine and uracil. So adenosine, guanosine, cytidine and uridine are the nucleosides of ribonucleosides. RNA, nucleosides of RNA. Chudandi, you have to remember the this place, substitution place. See this. What is the numbering? I will show you here. Here is. This is first carbon. This is second carbon. Ante kadamma, first carbon ani cheppanu kada nenu meeku. This is the first carbon. First carbon has right side OH alpha, left side OH beta. So beta carbon eppudu rasina kuda beta hydroxyl group on upside. Okay, this is beta form. This is also beta form of 2-DNA, 2-deoxyribose. See the numbering. Remember this numbering. This is the first carbon, second carbon, third carbon, fourth carbon and fifth carbon. And now apply this numbering in this. See this. Here This is the first carbon, right? And 1, 2, 3, 4, and this is the fifth carbon. Okay, Amma. And see this. The purine bases are linked to purine bases. These adenine and guanosine. Purine bases are linked to C1 of the saccharide, C1 of the sugar through N9. See this. See this, Amma. In the nitrogen base, the nitrogen number is 9. So here the combination between N9 and C1. This is called glycoside bond. This is glycoside bond. See, nucleosides are formed by the formation of linkage between C1 of the sugar through N9 of the nitrogen base. This is called glycosidic bond. అర్థమవుతుంది కదమ్మా? In the same way, in the deoxyribonucleic acids also. 1 minute. Structures ala unnai gaani very simple amma. Meer chudandi ikkada. This is first carbon and this nitrogen is, the numbering is 9. So here the bonding between this first carbon and 9 nitrogen. So this N9C1 linkage, this is called glycoside bond. Okay, and through this bond this nucleoside is formed. See here actually OH undalsindi ikkada, and this nitrogen should have hydrogen. Just a condensation process happened and the loss of water, and glycoside bond is formed between these sugar and the nitrogen base. Nitrogen base lo unna ee nitrogen, 9th nitrogen, sugar lo unnatvanti ee first carbon ki madhya bond form ayindi. Deenni glycosidic bond antam. Okay, so a combination of sugar and nitrogen base is called nucleosides. If you take adenine nitrogen base, the name is adenosine. If you take guanine nitrogen base, that is guanosine. If you take cytosine, that is cytidine. If you take uracil, that is uridine. This is about RNA, ribonucleosides. If you see in DNA nucleosides, deoxyribonucleosides, same, chudandi, but the difference is in the sugar. Kadamma, see this HOH on second carbon. Here only 2 hydrogens. This is deoxyribose sugar and these are nitrogen bases. How many types of nucleosides are there in RNA?
4.
4. And DNA? 4.
4.
Very good. So nucleic acids are composed of 4 types of nucleosides. Nitrogen bases 4 unnai kabatti 4 nucleosides vastayi, andulo emundi. But only the difference is deoxy అని ఇక్కడ మనం మెన్షన్ చేస్తున్నాం అంతే. అడినోసిన్, బట్ ఇన్ DNA deoxy adenosine. Here guanosine, deoxy guanosine. Cytidine, but in DNA ఏం చెప్తున్నాం? deoxy cytidine. And here uridine. One more difference. In the RNA, nitrogen base is uracil, but in the place of uracil, here we will take thymine. Okay, so this is called deoxythymidine nucleoside. Okay, amma? Nucleoside అయిపోయింది, combination of sugar and nitrogen base అయిపోయింది. Now we have to add one more group, that is phosphate. Okay, what is nucleotide? The combination of nitrogen base, sugar and phosphate. ఉన్నాయా మరి చూద్దాం. This is sugar and this is nitrogen base and this one is phosphate group. Okay, 3 components. There are 3 components. That is why this is a nucleotide. If you take RNA, what is the name? Ribonucleotides. If you take DNA, deoxyribonucleotides. The structures are very similar, but the difference is in the sugar. Here HOH is there in the RNA, but here only 2 hydrogens are there in the DNA sugar. Okay, see adenine, sugar and phosphate group. Okay, phosphate group is attached at which carbon?
5th.
Very good. C5. C5. 1, 2, 3, 4, 5. So C5 anamata. C5 దగ్గర ఏముండాల్సింది యాక్చువల్లీ OH కదా, అండ్ ఫాస్ఫేట్ గ్రూప్ H ప్లస్ అండ్ దిస్ OH కండెన్సేషన్ జరిగి వాటర్ వెళ్ళిపోతుంటే ఏం ఫామ్ అయింది ఇక్కడ? మళ్ళీ బాండ్ ఫామ్ అవుతుంది. చూడండి, ఫాస్ఫేట్ ఈజ్ లింక్డ్ ఎట్ C5 carbon. So this is the combination of nucleotide, phosphate, sugar and nitrogen base. If you take this adenine, what is the name of this nucleotide? Chudandi, 5-adenylic acid. 5 adenylic acid. This is fifth carbon kadamma. So fifth ante veelu ela icharu ikkada numbering ante fifth carbon has a phosphate group. That is why maybe the name is 5-adenylic acid, or you can say adenosine 5-phosphate. So fifth carbon has phosphate group. That is why the name is adenosine 5-phosphate. మీరు చాలా సార్లు చూసుంటారు AMP, GMP, UMP అని చెప్పేసి మెటబాలిక్ పాత్వేస్ లలో. AMP, what is this AMP? Can you guess? చెప్పండి, what is this AMP?
Adenosine monophosphate.
Very good, adenosine monophosphate. Here we are seeing only one phosphate group and the name is adenosine monophosphate, and actual name is adenosine 5-phosphate. Okay, and if you take this guanine, what is the name? 5-guanylic acid or guanosine 5-phosphate, like this. So or we can call it as guanosine monophosphate. Just ivanni practice cheyalamma meer. Okay, and then if you take this cytidine, what is the name? Cytidylic acid or cytidine 5-phosphate, CMP. If you take uracil, uridylic acid or uridine 5-phosphate, UMP. Okay, these are the names or structures of ribonucleotides. And if you see in the case of deoxyribonucleotides, similar, just the difference is deoxy. Structure anta kuda same untundi except this C2 carbon. చూడండి అమ్మా, అర్థం అవుతుంది కదా మీకు. In the RNA ribose, this second carbon has one hydrogen and one OH. But in the DNA, in the deoxyribose, second carbon has 2 hydrogens. There is no OH group. కాబట్టి మనం ఏమన్నాం? deoxyribose అంటున్నాం. So the name is deoxy. This is adenine. That is why deoxyadenosine 5-phosphate. This is the combination of adenine, deoxyribose and phosphate group. That is why the name is deoxyadenosine 5-phosphate or 5-deoxyadenylic acid. AMP same undi, but deoxy kabatti just ikkada em antaru, denoted by small d. Deoxyadenosine 5-phosphate, and this one deoxyguanosine 5-phosphate. Okay namma, deoxycytosine 5-phosphate, deoxythymidine, because in the DNA, in the uracil place, what will be there? Thymine. So the name is deoxythymidine 5-phosphate. You have to practice the chemical structures of these nucleosides and nucleotides. Okay, ఎందుకంటే రియాక్షన్స్ ఉంటాయి, మనకి ప్రిపరేషన్స్ ఉంటాయి కాబట్టి you have to practice these structures. How many types of nucleic acids are there?
2 types.
2 types. Names are?
Ribonucleic acid, deoxyribonucleic acid.
Where is the location of DNA?
Nucleus.
Chromosomes.
Yeah, chromosomes. Chromosomes ఎక్కడ ఉంటాయి మరి?
Nucleus.
Nucleus. Chromosomes are made up of
Nucleic acids and histone. DNA and histone proteins.
Very good. Just you are confusing, that's it. Chromosomes are consist of DNA and histone proteins. Okay. RNA is located
Cytoplasm.
Very good. Cytoplasm. It is not present in the nuclei. RNA ekkada untundi? Nuclei lo undadu. But it is produced in nuclei. But it is located in the cytoplasm. Okay. What are nucleic acids? Definition.
Polymer chains of nucleotides.
Very good. Polymer chains of nucleotides is called nucleic acids. What is the composition of a nucleotide?
Nitrogen base, sugar and phosphate group.
Okay. How many nitrogen bases are there in RNA?
4.
Names?
Adenine,
guanine, cytosine, uracil.
Very good. You have to remember this 4th one. Okay. DNA is composed of adenine,
Guanine,
thymine.
thymine. Only difference is thymine and uracil. RNA lo uracil untundi, DNA lo thymine. మిగిలినవన్నీ కూడా same to same. Okay. Adenine and guanine are derivatives of
Purine.
Metazole and pyrimidine rings.
Purine derivatives.
What I am asking?
Purine.
Adenine and guanine are derivatives of
Purine.
Purine.
Okay. And what are the names of pyrimidine derivatives?
Thymine, cytosine,
Thymine,
uracil.
uracil.
Just cut. Remember. Okay. కట్ అని గుర్తుపెట్టుకున్నారు అనుకోండి, ఒకవేళ మీకు మర్చిపోతే కట్ అని గుర్తుండగానే మనకి పిరిమిడిన్ డెరివేటివ్స్. సి అనగానే సైటోసిన్, యు యురాసిల్, టి థైమిన్. Adenine and guanine are purine derivatives. Cytosine, thymine, and uracil are pyrimidine derivatives. Next, how you can distinguish RNA and DNA? Sugars.
Oxygen atom.
Presence of oxygen.
Yes. There is only one difference. Ribose has hydroxyl group at second position, second carbon, but in the deoxyribose, only hydrogen atom is present at the second carbon. There is no oxygen at second carbon. This is the difference. That is why the name is 2-deoxyribose. Next, which ionomer is present in the nucleic acids?
Beta.
Beta ionomer. Very good. Ribose sugars are exist in dash form in the nucleic acids. Ante emanta ionomeric form. Beta ionomeric form. మనకు న్యూక్లిక్ యాసిడ్స్ న్యూక్లియోటైడ్ చైన్ ఉంటుంది కదమ్మా. ఆ న్యూక్లియోటైడ్ చైన్ లో ఈ న్యూక్లిక్ యాసిడ్స్, ఈ రైబోస్ షుగర్, డియాక్సిరైబోస్ షుగర్స్ లో OH గ్రూప్ ఎటువైపు ఉంటుంది? బీటా సైడ్, బీటా డైరెక్షన్ అంటే అప్ సైడ్ ఆ అరేంజ్మెంట్ లో ఉంటుంది. దట్ ఈస్ వై బీటా ionomeric form అని రాయాలి. దే ఎగ్జిస్ట్ ఇన్ బీటా ionomeric form. What is a nucleoside?
Hydrogen base
Sugar.
Sugar plus
Nitrogen
sugar.
base and sugar. Just
sugar.
two components. Nitrogen base and sugar. Can you identify the positions of glycosidic bonds in purine based nucleosides? Annar anukondi. Purine based
C1
nucleosides.
and N9.
Very good.
N9.
C1
C9
and
and
N9. In the pyrimidine,
Ma'am, that is N1 or N3, ma'am.
1, 2, 3, 4. Yes, you are right. One numbering, because we are going through substituents direction. If you go like this, there is no substituent. Generally, numbering kuda manam eppudu ekku ela istamo, more number of substituents na side istam. So here we are going anti-clockwise. 1, 2, 3, 4, 5. This is right. Right, you are right. 1, 2, 3, 4. 4 amino kada. So N1 and C1, right? N1 and C1. And here also 1, 2, 3, 4. N1 and C1. Okay, ma? This is glycosidic bond. Okay. Next, what is nucleotide combination of three components? Nitrogen base, sugar and
sugar phosphate.
okay. Phosphate group is joined at which carbon? C5 carbon.
Carbon.
So that is why the name is 5-adenylate acid, 5-guanylic acid. Ila rastu potunnam. Phosphate group is attached at 5th carbon. Okay. One question. Okay, time good అయిపోయింది. Retrosynthetic analysis. Do you have any idea about this?
Yes, ma'am.
చెప్పండమ్మా.
We will go in a reverse manner, ma'am, to synthesize our product.
Very good, very good.
We'll start from the product.
Yeah, start from the product. Okay, I am giving this compound. What is the name? 2-cyclohexyl ethanol. This one first carbon, this is second carbon. Okay, this second carbon has cyclohexyl substituent. So the name is 2-cyclohexyl ethanol. Okay, ఇది దీని పేరు. 2-cyclohexyl ethanol ప్రిపేర్ చేయాలంటే మనము అదే we have to go through this retrosynthetic way. So then what are the expected starting materials అనేది మీరు చెప్పండి, గెస్ చేయండి. విచ్ టైప్ ఆఫ్ రియాక్షన్స్ వీ కెన్ ఫాలో టు ప్రిపేర్ దిస్ కాంపౌండ్. జస్ట్ ఊరికే మీరు ట్రై చేయండి, వస్తే వచ్చింది, లేకపోతే రేపు నేను ఎక్స్ప్లెయిన్ చేస్తాను. బికాస్ జస్ట్ వీ హ్యావ్ టు నో అబౌట్ ద బేసిక్ కాన్సెప్ట్ ఆఫ్ దిస్ retrosynthetic analysis. Then based on this approach we are going to study how to synthesize nucleic acids. But you know, nucleic acids are large complex type of structures kada. They are complex molecules. అవును కదమ్మా, పాలిమర్, న్యూక్లియోటైడ్ పాలిమర్స్ కదా. న్యూక్లిక్ యాసిడ్స్ అంటే ఏంటి? పాలిమర్స్ ఆఫ్ న్యూక్లియోటైడ్స్. సో హౌ టు సింథసైజ్ దీస్ న్యూక్లిక్ యాసిడ్స్? సో వీ హ్యావ్ టు ఫాలో దిస్ retrosynthetic process. So we are going to see that retrosynthetic process and how to prepare nucleic acids through this retrosynthetic process tomorrow. Okay amma, మళ్ళీ మనకి Thursday, Friday, Saturday. Thursday, okay, see you on Thursday. Okay, మీరందరూ చాలా యాక్టివ్ గా ఉన్నారు. Keep it up. Very good.
Ma'am, are you in college, ma'am?
Yes, you can see
Can
my
you show the
background. Yeah, you can see my background. See.
Yes, ma'am.